Hi, I'm Evan Crummell, President and CEO of Protein-Flytics. Thank you for coming to our poster, Novel Assay Methods for Cancer Patient-Derived Organoids. This video will be a short presentation on the data presented in the poster. If you'd like more information, please download the poster itself, or you can contact me at ecrummell@proteinflytics.com. So let me now switch over to the presentation. Great. I want to start by acknowledging the co-authors and other people who helped out on this work. First, at Tulane University School of Medicine, Marguerite Metwison, who's an MD PhD candidate there, and her professor, Matthew Burrow. They did the bulk of the work developing the cell lines and some very exciting work with patient-derived organoids going down in their laboratories at Tulane University. Elect Vice's Oksana Serenko, who did the imaging and analysis of the PDOs that you'll see, Michelle Long, Protein-Flytics, and Glauco Suko, Griner Bio1. And as I mentioned, if you want more information, please contact me. So disease modeling with PDOs is a very exciting area, and a lot of labs are now getting involved in this. Very promising for not only understanding cancer, but moving into precision medicine, where you can now derive therapies based on responsive patient-derived tumor models. PDOs are generated from tumor biopsies, sometimes through mice and xenografts, and then into PDO culture where they can form organoids. It's really a nice translational tool and provides you a physiologically relevant system to evaluate tumor biology. But as a lot of new technologies, there's a need to automate a lot of these steps. It's a very manual process right now. So we have developed the Pu.MA assay system to do automated organoid assays. In this case, organoids and reagents are loaded into Flowchips, which I'll describe later. Those Flowchips are placed in a Pu.MA system located inside an incubator where all the assay steps are run. It's a very intuitive operation where protocols are preloaded. You just select protocol, press play. When the assay is finished, you take the plate out and then read it on your analysis systems, whether it be a plate reader or in this case, the Icon imaging systems. The instrumentation I described is the Pu.MA system already. And then the other really important piece of information here, instrumentation, is Immunofluorescence microconfocal imaging system. Very high resolution, very nice imaging system, all controlled by the MedExpress software. The Flowchips have been developed over the past several years. And while they look like a 384L plate, it's actually a lot more than that. Each chip has 8 lanes. Each lane is one test. And in each lane, there's a sample well in the middle. And here you can see a spheroid placed at the bottom of that sample well. Each sample well is connected to multiple reservoirs, so you can do media exchanges, staining, conditioned media sampling, etc., all done automatically through these microfluidic channels which connect the different reservoirs. A key aspect of our flow chip is this protected sample chamber. And this little cartoon shows the location and the utility of that. So organoids are placed in the bottom of this chamber. The chamber volume is about 1 microliter compared to the total volume of the well, which is about 20 microliter. So when you do reagent exchanges in and out, you exchange about 95% of the fluid, but you're not drying out your organoids. So it's protected from the fluid flows. And that also allows you to drain the complete media if you need to remove all your media from the sample well. So let me now get into the data on the PDOs, the formation imaging, and some of the disease modeling we did. These were TU-BCX4IC cells derived from our primary tumor, a patient down at Tulane University. This is a metaplastic breast cancer with a triple negative breast cancer subtype. You can read more about these in Marguerite's publication shown here, or some of the other publications out of Matt's lab. This particular tumor exhibited resistance to chemotherapy, and specifically paclitaxel. When we received the cells, they're cultured 4IC cells. We dispensed them 2,000 to 4,000 cells per well into a round bottom, low attachment plate from Corning. After about 72 hours, they formed nice round spheroids and they were ready for the assay. To facilitate the placing of the spheroids and doing the analysis, we coated them with nanoshells. So this is a novel material from Griner Bio1. These are magnetic nanoparticles that attach to the cell. And then this allows you then use magnets to hold and position them in place. And so the unique thing about this device here is now you can place these spheroids and put them in the center of the well, which really facilitates the ability to image them. Also, the nanoshell has been shown not to interfere with any of the biology, and it doesn't interfere with the imaging. So it's a very useful tool for improving your assay performance. The assay steps here, we would, with the organoids placed in our flow chips, we'd do a compound incubation, typically 24 hours in this case, although you can go up to 72 hours. After the incubation of compounds, we remove that and add in the stain. This could be viability stains or immunofluorescence staining. If washes are required, specifically with immunofluorescence staining, you can remove the dyes or antibodies and add in doing multiple wash steps. Once all this is finished, which occurs inside the incubator, you remove the plate. You can sample supernatants if you want to look for secreted factors, conditioned media, or just put it onto your imaging system to do that image analysis. These are beautiful images, and a shout out to Oksana for this really nice work here. Shows immunofluorescence staining with E-cadherin and CD44. And you see the nice tight organoid here, control a healthy organoid or tumoroid with a high expression of E-cadherin. With the treatment, the E-cadherin is downregulated or destroyed, and then the tumor itself falls apart. You can see some live cells here as evidenced by the CD44 staining. Again, the romidepsin does a nice job of killing the tumoroid. So we can then do further disease modeling. In this case, we're showing treatment of tumoroids with different compounds. The top one is romidepsin, the bottom one is trametinib. With increasing compound, you get the prerequisite killing of the cells and disintegration of your organoids, in this case using viability staining. So to analyze the spheroids, then we use MetaQuest software here using custom module editor, where you can isolate the individual organoids and then inside there do a cell-by-cell analysis. In this case, identifying live cells by calcium AM, they're shown in blue, and dead cells by aethanium homodimer, which are shown in yellow. With that analysis, you can get nice concentration response curves, and here for the 4 different compounds we treated them with. Interestingly, here we saw paclitaxel have very high EC50 value, about a factor 100 more than we would normally see with cancer spheroids. And that was very consistent with the patient's tumor response, which was resistant to Taxol therapy. So a nice validation of this method for doing disease modeling. So in summary, we've developed this Pu.MA system, which allows you to automate your organoid assays. We've shown its utility for doing patient-derived organoids and facilitating the ability to treat and image these organoids. It works with patient-derived organoids as well as different organoid types, and automates a lot of the common assay steps that you would be doing with these type of models, whether it be media exchanges, supernatant sampling, staining, etc. So again, thank you for coming to our poster and paying attention, and please download it and contact me if you have any more questions or would like more information.